Okay, so we've done transcription, and now the second portion of the central dogma is translation. So, if you recall, transcription, all of this, and if we're talking, whoops, sorry, if we're talking about, if we're talking about a eukaryotic cell, all of this here happens in the cell membrane, excuse me, the nucleus. All of this is in the nucleus in a eukaryotic cell. In a prokaryotic cell, there's no nucleus, so all of this happens just real close together. Whereas the RNA will then travel outside of the nucleus and try to find a ribosome, which may be free floating or it may be associated with the rough endoplasmic reticulum. And the ribosome will then use the information that is being delivered by the RNA, the mRNA, to make the protein. So we call this process translation. Okay, so what I'm going to do is, we're going to go through that process in its entirety. All right. So the first thing is the ingredients. You need to know all of the things, just like we did before, the proteins that are involved. You also need specific items for this as well. And what I didn't mention in the last lecture, which I probably should have, was that it's not just the proteins that are involved. You do need the nucleic acid bits, or the nucleotides specifically, that are going to be used or making up ultimately the messenger RNA. Okay, so you need those components as well. And we did talk about them. I just didn't say that outright. So here with my translation, I do need a few things. I do need my messenger RNA, okay? If you're talking about a prokaryotic cell, this came right from the DNA. DNA was transcribed, you made messenger RNA. Whereas if you're talking about a eukaryotic cell, then you're talking about the first thing that was made right off the DNA was primary transcript, or pre-mRNA, and then you have RNA processing. And that processing included the poly-A tail and the cap, and the removal of the introns, the splicing together of the exons. All of that was there before you got the messenger RNA. So the other things that you need to include would be initiation factors, and these are going to be proteins that are going to exactly what they sound like. They're going to initiate the process of translation once the messenger RNA gets out into the cytoplasm. Again, talking about a eukaryotic cell. Once it's exited the nucleus, then these initiation factors are going to be proteins that do exactly what they do. It sounds like they initiate the process of translation. Elongation are going to initiate the process of elongation during translation, which is going to build the protein amino acid by amino acid by amino acid. So it's going to create those chains. Release factors are going to stop the process. You have these enzymes right here called amino-acyl tRNA synthetases, okay? And I'll talk about what they do in a little more detail, but briefly what they'll do is they'll bind the appropriate tRNA, which are going to be right here and these things in purple, to the appropriate amino acid, okay? So there are very specific enzymes. For every tRNA that exists, there is a specific amino-acyl tRNA synthetase that will bind the appropriate amino acid to it. And then, of course, the ribosome. And the ribosome is made up of two different components. It's the large subunit and the small subunit. So this is the large, this is the small. And these both are made from the third type of RNA, because you learn messenger RNA. Transfer RNA is the second type in this lecture, and then rRNA, or ribosomal RNA, is the third type. So they're going to be making up the ribosomes along with proteins, ribosomal proteins, that ultimately make both the large and the small components. Okay? So I have my ribosome. We're looking up and close and personal. Again, in a prokaryotic cell, the ribosomes are just there. They're amongst the rest of everything that is in the bacterial cell, because there's no membrane-bound organisms, there's no nucleus. But the ribosome in a eukaryotic cell is going to be necessarily outside of the of the nucleus, either in the cytoplasm, or you do have the fact that, or there is the fact that both chloroplasts and mitochondria have their own set of ribosomes as well, which look awfully similar. In fact, they're very similar, if not identical, to the ribosomes of prokaryotic cells, which is another piece of support for the endosymbiotic theory. But anyway, that's neither here nor there. So I have my large subunit, and I have my large subunit, and I have my small subunit, and I've got these three sites: EPA, 8, APE, 8 backwards, and it goes in this direction. Okay, so this is how you're going to actually read it. So the amino acyl site, the peptidal site, and the exit site. All right, that's APE specifically. The amino acyl site will be the site that receives the first, or that receives the tRNA, okay, that's bound to the appropriate amino acid. This tRNA is going to enter the amino acyl site, okay, and when it does enter that amino acyl site, this tRNA is going to drop off this amino acid onto the amino acid chain that's already growing here, okay. So there's a process. We'll go through the sequence, but this tRNA will come in and drop off its tRNA, and that's the peptidal site is where the bond, that peptide bond, is formed between the former amino acid here and this one here. So it's going to create that peptide bond. And then the exit site is going to be where the tRNA itself. So the tRNA will leave from the exit site, will leave from the exit site and to go off and to rebind with another amino acid. Okay? Again, we'll go this step by step, so don't get too flustered by this. All right? So let's talk. This is your tRNA. So let's get a closer look at your tRNA. Transfer RNA. It is, remember, RNA is single-stranded, but RNA is also, it's or can create three-dimensional structures on its own. So again, this is one strand, but it can fold in on itself because of the nitrogenous bases interactions, as you can see. So C to G here, G to C, so on and so forth, A to U, U to A. These interactions that are forming, I hope you remember, the things that hold these together, in fact, hydrogen bonds, will create what we call a conformation or a three-dimensional structure for tRNA. And the tRNA flattened out looks like this, but it kind of looks like what they call the hairpin structure right here, in actuality in 3 dimensions. All right, so but do recognize, here's that green loop is right here. Okay, if you're colorblind, I apologize, but here's this green loop, which is located right here. Here's the red loop, okay, right here, and the anticodon loop is right here. Okay. So these are what corresponds with what where. Okay. Now, what is interesting is, or what else is important here is this amino acids attachment site right here. So this portion right here of the tRNA, this little portion, this tail right here, these are the same, are going to be attached to an amino acid. Okay. And remember, there are 20 different amino acids, so, but that is specific. It's not going to be to just any amino acid. And as a matter of fact, what's going to inform the appropriate amino acid will be this right here, this idea of an anticodon, okay? We'll talk about codons and anticodons in a second, but the anticodon is going to dictate, it's going to inform what goes here. And actually, it's the enzyme itself that will pick this whole tRNA up. And it's only going to bind with the appropriate tRNA that has this anticodon. All right, so the tRNA synthetase is anticodon specific as well as amino acid specific. So they're built specifically to recognize this anticodon and to attach this particular amino acid, whatever it happens to be, to this tRNA with this anticodon. Okay, so it's very specific. That way, this message, okay, this message right here, is going to be translated into appropriate amino acid sequence. Okay? Okay. So let's continue on. All right, so this is what I'm talking about, what I just finished talking about. Here are my free amino acids. This is glycine, valine, isoleucine, and I've got the what are called uncharged tRNA. Okay, so these represent my tRNA. Here's the anticodon. Here's where the amino acid's going to attach. Anticodon, amino acid will attach here. Anticodon, amino acid will attach here. Okay, so these are uncharged. When I say uncharged, that means they have not been bound to an amino acid. Okay, this amino acyl tRNA synthetase, all right, this specific one right here is always going to pick up the tRNA that is UAG, okay, and will always bind this particular tRNA to the isoleucine. Okay, ooh, sorry, the isoleucine amino acid. So always, it's very specific. This blue one will always pick up a tRNA with a UAG anticodon and bind it to an isoleucine amino acid. Boom. All right, always. And then down here, in this particular amino acyl tRNA synthetase, will bind to tRNAs that have this particular anticodon and bind it specifically to this particular amino acid. So as you can see, I have multiple tRNAs that are going to bind the appropriate amino acid to the tRNA. Okay? So each of it, yeah, blah, 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 blah. And the enzyme attaches the amino acid to the 3' end of the tRNA. Okay? And once I do that, I become charged. And now this can go find itself to some ribosome that is building proteins. Okay? Okay. So this is sort of the summary of what I'm just talking about. I've got my, we did this yesterday, the transcription, non-template, the template. This will undergo transcription with all the appropriate enzymes and nucleotides, and I will build ultimately this right here, which is my messenger RNA, okay? And this messenger RNA will then be translated into an amino acid sequence. So, we said anticodon, here's this tRNA right here. Here's that tRNA with it's charged because here's the amino acid and here's the anticodon. The anticodon exists on tRNA, a codon exists on messenger RNA, okay? So messenger RNA has codons, transfer RNAs have anticodons. And they're basically opposites, or complements, I should say. And this codon right here, AUG, adenine, uracil, guanine, okay, is a really important codon, and you do need to know this one. This one codes for the amino acid methionine, okay? And it is also called the start codon. Because this right here is where the ribosome will start to build the proteins and everything that happens downstream of it, everything that happens afterwards, all right? Actually, I think it's the other way. Yeah, because it goes in 3 to 5, so it's going to be down in this direction. But that's the starting point, is AUG, and then the ribosome will bring in the appropriate tRNA with the appropriate anticodon, and it's charged with the appropriate amino acid. So as you can see, here's the anticodon. AUG is anticodon to A to U, U to A, G to C. And so there I have the anticodon binding to, briefly, recognizing the appropriate codon, and therefore this will then drop off this amino acid and start the chain. So all proteins in your body, they're being translated and transcribed and translated. All proteins are going to start with the amino acid methionine. What happens after that is completely dependent on what's happening downstream of that codon. All right, and a codon, in case I was not clear about this, a codon is 3 nucleotide pairs, or I should say bases. So they're triplets, triplet of nucleotides. And the next one would be whatever is here, you know, and the next one is the next three, next three, next three, next three. There's no punctuation, there's no pausing, there's nothing. It just reads until I get to the end, or an exit, a termination sequence, okay? So this right here, ladies and gentlemen, is super important. No, you don't have to memorize this. I will give it to you on the exam, but you do know how to read this. You know how to read this. So first position, the 5' end, second position, third position. So it's 5 to 3. I think I had it right the first time. So it's back here. Let me see. Make sure that no one's confused. So right here, 5 to 3. So it's red in this direction. So yeah, it's AUG. So this is the next. This would be. Whoops. Sorry. This would be the first codon right here. The next codon would be these three. The next codon would be these three, and then those three, and then those three, and then those three, and so on and so forth until you get the end. Okay. So here's my codon chart. Okay. First position. So that would have been the A for the first position, U for the second position, and then G for the third position for that codon. And here it is. Boom. AUG. All right. And that's methionine, or M, which is the start codon. Okay, so as you can see, I want to point out a couple of things here. The first thing I want to point out is that you have, you know there are 20 amino acids or so, but there is repetition in the code. So in other words, these right here, all of these code for the same amino acid, leucine, okay? So there are multiple codons, triplet base pairs, or triplet bases that code for an amino acid, multiple ones. And that's the case with most everything except for methionine and one other, tryptophan. Yeah, tryptophan I think is the only one. Methionine and tryptophan are the only one where you don't have duplicate codons coding for the same amino acid. Okay. So what does that mean? Well, what it means is that in case there is a mistake in your DNA or anything that flows from the DNA, it's like also your messenger RNA, if there is a mistake, which we call a mutation. Let's say there was a mutation and the codon was supposed to be CUU, but the mutation ended up being CUC. As I can see down here, here's CUU, here's, cancel. Here's CUU, and here's CUC. So if there is a mutation and I do mess up that last base pair, or the last base, then nothing happens. I'm still getting leucine in that specific location where that codon is in the sequence. So it doesn't really matter. And that is actually pretty valuable. But what happens is these mutations don't end up resulting in anything detrimental. They also don't result in anything that's more beneficial, but at least the protein that is produced from this particular mutation is still functional, and is still going to function in the same way. Okay, so this allows for mistakes to happen, so that there'll be less of a problem if there are in fact mutations. Okay, so do make sure you go over this. If I say, what is the amino acid that for which UUU codes for, then you find the first base, second base, third base. Here's UU, so it's phenylalanine. Okay. The other thing I want to point out to you is these right here, UAA, UAG, and you do need to know these, UGA. Okay, so you need to know AUG, and you need to know the stop codons. These are going to tell when that messenger RNA, when messenger RNA, or I should say, I'm sorry, when a ribosome runs into the sequence in a messenger RNA molecule, it will stop the process of translation. It will no longer do anything that's downstream of that codon, and the termination will occur. Okay, so you got to know those. Okay, let's see. That's your codon chart. Again, you don't need to memorize that. You may have to memorize some. You'll definitely have to memorize that in biochemistry, or I don't know if you'll have to memorize that. You'll have to know the structures of amino acids. You might have to memorize some of this in biochemistry. I don't think you have to memorize in cell biology either, but you do need to be familiar with it. You need to know how to use it. Okay, so process of translation in more detail. Again, here I have my messenger RNA, and I'm going to use that messenger RNA to bring in the appropriate transfer RNA that's charged with the appropriate amino acid into the ribosome so that you can build an amino acid sequence and thus ultimately make the protein that you're trying to make. Okay, so this is how we do this. And notice this word up here, monocistronic. Okay, monocistronic messenger RNA. This is going to be eukaryotic. Okay. And I'll talk about what this means soon, but just bear with me because it's better to do a comparison with the polycistronic and your prokaryotes. So initiation eukaryotes is at the 5' cap, and the first AUG is in the start codon. So here's the cap. Okay, this is what's going to be read first into the ribosome. And the ribosome is just going to read this until it gets to the start codon right here, AUG. Once it does that, then it will undergo translation and bring in the appropriate transfer RNAs that are charged, okay, into the ribosome that will ultimately use this information right here in this portion. These are all the exons to build a polypeptide, and will stop when it hits a stop codon. Okay, so I always think of, like, if you've ever been to an arcade or something like that, or have just seen this kind of process where you've got the stationary something, which isn't exactly accurate, but you got the stationary something and you win all these tickets, and you've got a ticket counter that will just kind of feed the tickets into this counter that will count the tickets for you. It's kind of like that motion. In my head, it's not necessarily that. It might be, it's the ribosomes can also move or will move versus the messenger RNA, but there you go. Okay, so let's polycistronic means that what you can have, so monocistronic is there's only one ribosome reading a messenger RNA at a time. Whereas polycistronic, which are again prokaryotes, is the idea that multiple ribosomes can be reading a single messenger RNA strand at the same time, and the messenger RNA itself will also, or can also, or does also have multiple genes. So remember, every green box here, this is all AUG. So every time you run into those AUG codons, that's time to translate and build another amino acid sequence. So this can happen multiple times on multiple, again, so it's multiple ribosomes making multiple copies of multiple genes on a single mRNA strand. Eukaryotic cells, it's one, it's going to be one gene. You can have multiple ribosomes actually reading the messenger RNA, but there's only one gene on them. It's only one thing. You're not going to have multiple methionines on it. Okay. I should put it that way. I think I said it wrong earlier. But again, so in either case, multiple ribosomes can be reading it. But in prokaryotes, there are multiple genes on a single mRNA, where in eukaryotes, there's only one gene. Okay. Okay. So initiation. So this is what happens. This is out in the eukaryotic cell. So this is out in the cytosol, the cytoplasm, and this messenger RNA has come in, and here's the 5' cap, okay? And what I've got is a situation where the messenger RNA will will run into these initiation factors. So they're proteins that are on the lookout. They're feeling their way around. When they come in contact with the messenger RNA strand that hasn't been initiated yet, then they're going to attach to it. And they're going to attach to it in such a way that they're going to bring over with them the small subunit of the ribosome. So the ribosome, remember, there are two parts, and they're not always together. The small subunit is going to be alone, waiting for the initiation factors to recruit it to newly exposed messenger RNA from the nucleus. Then my tRNA is also going to be involved in this, because the tRNA is going to, with the appropriate amino acid, so it's going to be methionine. It's always methionine. You always have to have this tRNA with UAC first, that's attached to methionine first. So all of these things get together, all right, and you make what's called an initiation complex, okay? And the complex is going to be the initiation factors, the charged tRNA with methionine, and the small subunit, okay? That will ultimately be part of it, the initiation factor, or those things. And then what I get next is, once that's all created or all together, I'm going to bring in the large subunit as well, okay? So the large subunit is the last thing to come to be, or come and join this complex, okay? And what happens is it gets placed on, and that large subunit will have the appropriate EPA sites. So, but it gets placed on top of the messenger RNA with the tRNA now inside of the ribosome. Because remember, the tRNA was already there. It was held in place by the initiation factors and hydrogen bonds. And so the ribosome then gets the large subunit of the ribosome just claps on everything, and it covers up the tRNA with the methionine, okay? So the next thing that happens is the ribosome will read the next, let me choose a different color. We'll read the next codon on the messenger RNA and bring in the appropriate tRNA with the appropriate anticodon into the amino acyl site. And that, remember, that tRNA is also charged, okay? Okay? So, once the amino acyl site is empty, a new tRNA with the appropriate anticodon to the new codon that's being exposed will enter in this site, the amino acyl site, with its appropriate amino acid, okay? And then with these two tRNAs in the appropriate locations, all right, the polypeptide or the peptidyl site will help initiate or form a peptide bond, which is right there, between these two amino acids, okay? So the next thing that happens is all of these move down, okay, they shift. And actually what happens is the ribosome actually moves in this direction. And when the ribosome moves in this direction, this tRNA ends up in this site, all right? So you can see that in your head, as if this is moving in this direction down the messenger RNA, then these actually stay in place, but ultimately will end up in the E site. This second one will end up in the P site, so it moves here. And then the A site is now open, it's empty. And now I can repeat the process. I can bring in the appropriate charged amino acid with the appropriate anticodon for this next codon right here, and bind them up and pair them up, okay? That way, the next amino acid in the sequence will then create a bond with valine, and so on and so forth, okay? So when we do that, again, the UAC ended up in the appropriate in the appropriate exit site and does just that. It exits, and now it's no longer charged. My peptidal site has, the P site has the former tRNA that was in the aminoacyl site, and now a new charged tRNA with the appropriate anticodon is going to come into the aminoacyl site, bind to the codons, and drop off its amino acid to valine, okay? So which would be ARG. This will go on, and we'll search for a new aminoacyl tRNA synthetase okay. I'm having trouble. Okay. Synthetase to charge it again. Okay. It's specific aminoacyl tRNA synthetase to charge it again. Okay. So again, this is kind of like Pac-Man in that ribosome is going to move to the right in this particular figure. It's going to move to the right, and every time it moves to the right, the tRNAs don't move anywhere relative to where they are. They're going to stay on the codon. They're going to stay attached to the codon of the messenger RNA that they're complements with, and now those new sites will have a different function, because they're going to be in a different function or a different site. The tRNAs will be in a different site. So the E site will exit. The P site will create a new peptide bond between the between the last amino acid that was dropped off and the new one, and so on and so forth. Okay. So again, that's what you see. Now that this charged tRNA is in the this is right here. Now that this goes in here, drops off its amino acid, binds it to valine in this particular case, and then what happens after this? I hope you can see in your head that this will move in this direction, these two will move in this direction, this will end up in the E site, this will end up in the P site, the A site then opens up, and whatever this codon is will initiate bringing in a new tRNA with the appropriate amino acid, and so on and so forth. And I keep doing this until ultimately I hit somewhere down here, one of the stop codons. All right? Wherever that may be. When I do hit the stop codon, okay, which in this, again, we know it's UAA, then that stop codon, that tRNA that comes in, it's a little more complicated than that, but we call this a release factor. And what happens is this release factor will undergo hydrolysis, or will hydrolyze the bond that is connecting the last amino acid to that last tRNA, so that this can then be its own entity free. Okay. So again, yeah, so this is hydrolysis. This release factor hydrolyzes the bond that was between this tRNA and this amino acid, and releases the amino acid, this polypeptide, into the cell, so that other things can happen after that. Then after that, so this is called termination. So after that, you get the breakdown of this entire complex, and the recycling of this messenger RNA into other components, and these continue to go back and become recharged, and everything starts all over again. Okay? So, in case I was not clear, initiation, elongation, termination. Those are the three components. Initiation, bringing the initiation factors with the tRNA that has methionine attached to it, and the small subunit, and then the last thing is the large subunit. All of that is initiation. Elongation is the moving of these, of the ribosome down the messenger RNA, and bringing in the appropriate tRNAs with the appropriate anticodons for the codons that are there, dropping off the amino acids, and as that happens, the amino acid chain continues to grow. And I do this until I hit a stop codon. Well, termination will happen. I get the hydrolysis, of that last amino acid from its tRNA and the breakdown of this entire complex. And everything gets recycled to do it again, okay? So this is just a reminder right here, that, again, why you need to know these methionine AUG and then UAA, UAG, and UGA, okay, as important ones. The rest of these, you should be able to, again, read this, because I very well may give you a question on your test or a quiz that asks you to translate or transcribe something, okay? So just bear that in mind. And again, these are codons that you're reading. Codon. This is a codon chart, and codons are in messenger RNA, okay? I could just as easily give you a tRNA sequence, all right? And if you use that tRNA sequence to try to put together anything from this chart, you're going to get it completely wrong, because this is a codon chart, not an anticodon chart. So you need to make sure before you start reading this chart and applying it to something that you have, in fact, the codons in front of you, not the DNA and not the anticodons, okay? Okay. We talked about this a little bit with the three-dimensional structures and the protein folding domains. In this particular case, what I'm talking about is remember, these are quaternary structures at this point. I've got multiple tertiary structures, and the domains themselves are going to be elements or portions of the protein that have everything to do with their function. So as you can see here, I've got different colors associated with this right here. So all of these alpha helices right here, this one domain, and then my other stretches, either beta pleated sheets or just stretches of the amino acids that have a different function or attached to something different if they are in fact going to do that, etc., etc. Okay, so that's all you really need to know about that. Evolution of proteins. So here's where I told you, change to the genetic code is a mutation. A mutation is a problem, or can be a problem, when you're supposed to have an amino acid in a specific position like phenylalanine, and because of mutation, instead you get something like isoleucine. Remember, the R groups are going to what gives the characteristics to these amino acids, and if those R groups need to say be polar, they need to be polar because their particular location is going to in the amino acid sequence will create a point where you've got a three-dimensional structure, appropriate a three-dimensional structure. If isoleucine, so this is nonpolar. So they're both nonpolar, so it might not be a problem, but it also might be a problem if I get a polar substance or a basic substance or an acidic substance, and when I say substance, I mean R group. So those characteristics, they have to be appropriate. They have to be give the same kind of chemical properties, or have the same kind of chemical properties, so that the three-dimensional structure of the protein is what it's supposed to be. As soon as you get amino acids in the wrong location, and some are more detrimental than others, but as soon as you get that, then you get a different protein with maybe a different function, or just non-functioning protein entirely, and that may or may not be good. Usually when you're changing the shape of a protein, it's not going to be good. Usually it won't be good, because that protein has a very specific function, and without the shape, it's not going to have the function. It's not going to do the function. Okay, so if I keep going, if I go back here, this would be equivalent to my protein. So gone would be the amino acids that need to be in that particular sequence. All right, I need those 4 amino acids, G-O-N-E, in their specific sequence in order to achieve a functional protein. Now, I'm not saying that only 4 amino acids are going to make a functional protein. What I'm saying is, what I'm saying is that the sequence needs to be appropriate in whatever sequence we're talking about in order for it to achieve that specific conformation or shape. So what can happen is I can definitely create a bunch of nonsense. All right, all of these are nonsensical. These don't make any sense. They don't translate into anything, and they won't be selected for usually. Okay, whereas what I can do is I can definitely have mutations that creates a something that makes sense. At least it looks like a functional protein. It may not have the same function, it very well may, but it may not have the same function, but at least it is a protein. And sometimes, on occasion, that these new proteins that are made are actually going to be more beneficial. Okay? So they could be detrimental or deleterious. I'm having trouble up here. Deleterious. They may be neutral. They may not have any differences in the protein function at all. So, we want gone, that's what was in the code, the genetic code, but none will work just fine. It doesn't really matter. Or it could be beneficial. Like it could add a element of benefit, or an additional benefit that is selected for over time. That doesn't happen very often, but it does happen. So just be aware of that's how these things could go. And be careful with the language. Don't say it's going to result in, or it will necessarily. Try not to use absolutes here, because that's the whole point. Is that a mutation may not result in anything. Like a mutation, if the amino acid, if the genetic sequence that coded for GONE was changed, it still very well might code for GONE. It might do that, because of the repetition and in the codon sequence. 6 different codons all code for the same amino acid. So you could get gone again. Or if you don't get gone, so mutation doesn't have to do anything. If it does do something, it might be neutral. It might be beneficial. It might be deleterious. All right. It's more often deleterious than beneficial, but it's more neutral than anything. All right. Most mutations are neutral. They don't result in anything. Because sometimes you can completely replace an amino acid and it makes no difference whatsoever. Because that particular amino acid isn't, like its position in the chain isn't what's really the keystone behind the whole shape. I mean, you could replace it, it's not a big deal. But sometimes it is, okay? So why are mutations eliminated in a population? Because, again, as I said, sometimes we call them silent. Sometimes they don't do anything bad. They just exist, all right? They just continue to be because they didn't do anything bad. So you may not have, and sometimes they are beneficial. And so if they are beneficial, then they may proliferate in the population, okay? So this is an overall summative thing. Transcription. You need to be able to describe this to me, all right, or to someone. Transcription starts with DNA, and let's stick with eukaryotic cells, starts with DNA. When the appropriate environmental conditions are there, then you get these transcription factors that come in and are going to bind to specific, and sometimes transcription factors don't have to be proteins. They can be steroids. They can be hormones. So sometimes hormones will come into the nucleus and act as transcription factors. They start the process of transcription by bringing in the appropriate proteins that are going to support this and that are going to ultimately make the transcription complex. Not translation complex, the transcription complex. So the transcription complex to include the be-all and end-all, the RNA polymerase. Your RNA polymerase is the big one that's there. It's going to be used to separate the DNA, and 2, build a messenger RNA strand. And in eukaryotes, remember, it's pre-mRNA, all right, or primary transcript. Those are synonymous. DNA is always read, the template strand is always read 3 to 5 prime. The messenger RNA is always built 5 to 3 prime, okay? Once that is created, then the RNA must be processed in eukaryotes, where you get the addition of a cap and a poly A tail, and the removal of introns, and the splicing together of exons, and then I get my messenger RNA strand, my actual, not pre-mRNA, but mRNA, that leaves the nucleus. And it leaves the nucleus through these nuclear pores, where it's going to go out and search for a ribosome. Once it hits the ribosome, then, or I should say, once it hits the cytoplasm, then there are several different features that are going to happen here. The initiation factors are going to bring the new messenger RNA or I should say, they're going to combine the new messenger RNA with the small subunit of the ribosome, along with the charged UAC with the methionine tRNA, and ultimately the last thing that happens is the large subunit gets, sealed on top, and then the ribosome reads down the messenger RNA, and you get, so that's initiation. Then elongation is, and remember, initiation, elongation, and termination all happen also in transcription. So those are, understand that you have initiation, elongation, and termination in transcription. You have initiation, elongation, and termination in translation. Those are different. Those are completely different. All right, so initiation here is all of those things coming together to make the ribosome and the messenger RNA with the appropriate tRNA. Elongation is the ribosomes reading down the messenger RNA, bringing in the appropriate tRNA, charged tRNAs, in the appropriate sites, EPA site, whatever site is appropriate. They come into the they come into the A, except for the first one, comes into the A, moves into the P, exits through the E, okay? Dropping off those amino acids until I get to a stop codon, where hydrolysis occurs between that tRNA and the amino acid, and now I've got this polypeptide that is probably going to be a subunit of some major protein, okay? That will cover translation. All right, it's a bit much, I understand, I do get it, but again, you got to get down with this. Just sit down and kind of write out the process of what's going on, who's involved, and that sort of thing, okay?